Good morning and good afternoon, everyone. Welcome to today's webinar entitled Get Closer to the Truth, Stellaris Confocal Reimagined, being presented today by Dr. Petra Haas and Dr. Julia Roberti of Leica Microsystems. My name is Andrea, I'm with Labroots, and I will be your moderator for today's event. Today's educational seminar is brought to you by Labroots and sponsored by Leica Microsystems. For more information on our sponsor, you can click on the sponsor logo on the left side of your screen or visit leica-microsystems.com. I would like to remind everyone that this event is interactive, so we encourage you to participate by submitting as many questions as you like at any time during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. The presenters will be answering as many questions as we have time for at the end of their presentation. You can also use this Ask a Question box to report any trouble you have in hearing the presentation, or you can also click on the Support tab for any help you might need, and that's found at the top right of the presentation window. I'd like to now introduce today's presenters, Dr. Petra Haas, Senior Product Manager, and Dr. Julia Roberti, Product Manager for Advanced Confocal Imaging, both of Leica Microsystems. For complete biographies of our speakers, I invite you to visit the Biography tab at the top of your screen. But with that, I'll go ahead and turn it over to our speakers to get things started.
Thank you so much. This is Julia speaking. It's great to be here today to open together a new chapter in Leica Confocal microscopy history. We will talk about innovation together with Petra, and we will talk about why we have pushed ourselves to make the best technology available to you for your research. And we started imagining how it would be if you could have the power to see more, the potential to discover more, and the productivity to do more. And in doing so, we have reimagined confocal microscopy. So you can acquire more accurate and reliable data to prove your hypothesis. You can extract a new dimension of information from every sample and increase the scientific impact of your research, and to allow you to set up complex experiments in a few clicks. And in a nutshell, we have reimagined confocal microscopy to get you closer to the truth. And so our new confocal microscope, Stellaris, was born. And we have two great products. We have Stellaris 5, which sets a new standard in confocal imaging, and we have Stellaris 8 to gain more knowledge about the specimen from different angles. Stellaris is built on three pillars and three attributes. Stellaris gives you the power to see more, to get and collect more accurate and reliable data. Stellaris also gives you the potential to discover more, and this is related to the access to additional dimensions of information from which you can get additional insights. And this is using lifetime-based information that allows you to explore the function of molecules in the cellular context. And finally, Stellaris gives you the productivity to do more. And this productivity means that you can set up an experiment in a few clicks instead of setting up a bunch of individual imaging parameters. And it's the ability to get great image quality at the speed you need for your results. And it's also the ability to navigate and locate the interesting regions in your sample and be very efficient in your workflow. So we will go in details about these attributes. So now I will hand over the presentation to Petra.
Thank you, Julia. So actually, I would like to start by telling you what is behind the power to see more. So why is it relevant to deliver more power? So first aspect of that is that more power gives you more brightness to see more details. So this also provides expanded multicolor flexibility, and it improves your gentle live cell imaging capabilities. How does STELLARIS give more brightness and more detail? So this already is a very nice multicolor image captured with a traditional confocal, and here on the right side is what you will get with STELLARIS. So STELLARIS allows you to detect even low abundance species at endogenous levels through its enhanced sensitivity, and you get quantitative results due to the proprietary new photon counting approach that I will explain a bit later. So one key innovation of the STELLARIS platform is the new Power Heidi detector family with its 3 members that I would like to introduce to you now. So Heidi S, which will be our new standard for STELLARIS, building on silicon-based technology, and we believe that it will actually set new standards due to its all-rounder capabilities. And there's the Heidi X. This is an excellent detector for functional imaging based on Leica GASP hybrid technology. And there is a new variant of the GASP-based detector, the Heidi R, which is especially suited for applications in the near-infrared range. So let's look in more detail, starting with the Power Heidi X. It is using silicon-based multi-pixel photon counter technology. It's actually, as you can see on this graphics, an array of individual detector cells, and a photocathode material is on top, which receives the incoming photons. They are converted into photoelectrons, and each cell here has its own avalanche diode that converts the single photoelectrons in charged pulses, which are recorded at the anode. And so despite being an array, all signals are wired together in one output. So what is behind this exceptional performance that we're going to demonstrate in the next few slides? So in addition to the detector chip, this chip is actually combined with Leica engineering, which includes proprietary electronics and optomechanics, and the same applies to the implementation of an efficient cooling scheme to suppress dark noise. So what do we do with the output signals that are coming from this detector? So for the charged pulses emerging from the detector, 2 independent pathways are available to process those signals, resulting in 2 offered operating modes. So first, there's the analog mode, which is suitable for high dynamic range imaging, and this means that it's very robust in terms of the signal levels that the detector can handle. And second, there's the photon counting mode, where extremely low dark noise allows to capture even faintest signals, and it makes it the choice for quantitative measurements. Also, it allows to perform fluorescence lifetime-based imaging that you will hear later about. So the other 2 members of the family are Heidi X and Heidi R, both deploying GASP-based hybrid technology. And the function of these detectors is depicted in the scheme here, where again we have a photocathode material on top, where incident light is collected and is converted into photoelectrons, and each individual photoelectron is being accelerated by a high voltage to the photon anode, where a charge pulse is generated and read out at the anode. So ideally, each incident photon is converted into 1 charged pulse emerging from that detector. And this is the basis for the photon counting capabilities of the Heidi X and Heidi R to deliver the imaging of low signals with extremely little dark noise. So ideally suited for quantification and again fluorescence lifetime-based imaging. So what are we doing with the photons that our Power Heidi detectors collect with STELLARIS systems? So if we look at how conventional photon counting works, you can see that if photons are coming sparsely, everything is good. So one photon delivers one signal basically. And so, but if photons arrive closer to each other, there is a danger that they cannot be resolved since they are below the pulse pair resolution of the detector. in this example, conventional counting would actually miss one count. So this is where the counting scheme from STELLARIS differs. Here, we're making use of pulse width information, and this gives us the chance to know if one photon or more photons actually triggered a given pulse. And this enables a better capability to count signals, and it also makes quantification better as well. So at low abundance imaging, conventional counting and power counting give the same results. At high signal intensities, power counting significantly enhances detection sensitivity and also the dynamic range. The visual that you see here shows the benefits of power counting. So signals coming from the detectors are shown as a function of the excitation intensity. In gray, you can see how conventional counting behaves. It increases in a linear manner at low levels, but then you can see the saturation behavior of the detector. And the benefit is that with power counting, you significantly increase the dynamic range of the detector, and also you increase the linearity. Here on this slide, you see a side-by-side comparison of images that were taken with conventional photon counting versus those taken with STELLARIS power counting. And you can appreciate the gain in image contrast. In particular, this is beneficial on images taken with a resonance scanner, where the time on each pixel is very short to acquire photons. So you are working normally in a regime where you benefit most from the power counting. So STELLARIS gives you also access to expanded multicolor flexibility. What is this good for? So if you look here on the left, you see an actin staining where a light is collected all the way up beyond 700 nanometers. This is what you can see on a traditional confocal system. With STELLARIS, you see much more. In this example, 2 additional markers in the red range of the spectrum are accessible. So you add basically 2 more markers beyond what is possible on a conventional microscope. And don't forget that the rest of the spectrum is still available to generate more context to answer your scientific questions. So you profit from a broader choice of markers due to both the extended excitation and also detection in the near-infrared range. So one innovation driver which allows for this is the next generation of white light lasers. So the white light laser as such emits, as its name suggests, white light composed of all available wavelengths. And the way how you basically pick the wavelengths you're interested in is facilitated by our proprietary technology, which is acousto-optical beam splitter, the AOBS. So the AOBS allows to pick up to 8 lasers simultaneously in nanometer precision that can be tailored to the optimal excitation wavelength of the fluorophores in your sample. So the Leica white light laser provides complete spectral freedom. And with STELLARIS, we offer 2 new variants of the white light laser. So on the STELLARIS 5, we cover a range from 485 nanometers to 685 nanometers, which allows to excite already a fair number of redshifted dyes. And STELLARIS 8 comes with a white light laser which goes from 440 nanometers to 790, so it extended both into the blue and into the red. So this laser is ideally suited both for lifetime-based imaging of CFP-based sensors, for example, as well as optimal tool to cover the whole visible and near-infrared spectrum. So how does STELLARIS improve your live cell imaging capabilities? This is basically enabled by the synergy of some of the key innovations I just introduced. So the Power Heidi family, the spectral freedom of the white light laser, and not to forget the fully redesigned optics for enhanced transmission capabilities. The image here, by the way, shows a configuration with 4 Heidi X detectors and 1 Heidi S, which is sitting in the space in between. So simultaneous 5-color live cell imaging is at your disposal. So with this, I would give back to Julia, who will now tell you more about the potential of Stellaris.
Thanks, Petra. So now let's move on, and I want to talk with you now about the potential of Stellaris to discover more. And as I already mentioned, this is related to the use of lifetime-based information to get new insights about your sample. So this has the potential to really increase the scientific impact of your research and your results. And we see this as that it will become really a transformation of how microscopy is done. So with this potential, you can explore a new dimension of information, and also it gives you the possibility to multiplex beyond the spectral options. So you can gain access to functional imaging, and we will see now in the next slide how we can achieve this. So the technology that is supporting the Stellaris potential has a name, and this name is TauSense. So TauSense is a set of tools based on fluorescence lifetime. It gives access to this powerful additional information, qualitative and semi-quantitative. And it's gate-based technology, and it's also average photon arrival times that are processed at the FPGA level. So when I talk about this exploring a new dimension, I want to show you what I mean with this example. So we have now here a very nice image of Arabidopsis, and this here on the left is what you would see when you do intensity-based measurements. So here we have the possibility of observing chloroplasts by the endogenous fluorescence. We also have actin that has been stained with light ACT Venus, and we have the membranes stained with propylium iodide. So I can get very nice structural details, but I don't get any more contrast, and it's fairly difficult to see where the membranes and where the chloroplasts are sitting. But with Stellaris here on the right side of the image, I get this additional information, and then I can identify the chloroplast and the membranes and the actin filaments, and this is all enabled by one of the TauSense tools, which is TauContrast. So I want to explain how TauContrast works now, and for this I will use as a basis a very simplified version of the Jablonski diagram, where I can see how the fluorescence process happens. So I have my molecules in ground state, and then they are excited, and they will emit fluorescence. And as an outcome of this process, I will have a fluorescence intensity that I can measure as a number of photons, and I will have a fluorescence lifetime. How can I measure the fluorescence lifetime? That is basically the average time that the molecules stay in this excited state. Well, we have our white light lasers that are pulsed laser source. So then I can use this laser source, and I can use the photon counting capabilities of our detectors to measure the time it takes for the photon to be detected. And in principle, I could, from this information, estimate the fluorescence lifetime of my system. But what I will do is for TauSense, I will not go all the way to fluorescence lifetime. I will use the information that comes from the photon arrival time of the photons emitted by my molecules. So for TauContrast, I have my number of photons, my fluorescence intensity, and I have the photon arrival time. So what Stellaris will do is it will take this information and it will process it at an FPGA level, pixel by pixel and on the fly. And the outcome of a TauContrast experiment will be a fluorescence intensity and in each pixel, the average photon arrival time. So to show you how it works for the example I show you, I have the pixels and I will have the intensity in each one. And I will also have color-coded the information of the photon arrival time. I will do this for the entire image, and with TauContrast, I get instantly this arrival time information with every image in live and in acquisition mode. And this information reflects the environment, the microenvironment of the molecule, and that gives me extra information. Another aspect of this additional information is the capability to separate species beyond the spectral options. And that, to illustrate that, I have another example. So I have here a sample that has been stained with light ACT and neon green that you see here on traditional or intensity-based measurement, as you see here on the left in yellow. I also have mitotracker green for the mitochondria, also here. And these two stains I detect in the green channel. And then I set a red channel in which I'm detecting a nuclear stain, nuc red, and sear tubulin. And these both dyes you see here in gray. And as you can see, with intensity information, I'm not able to distinguish really where the actin and where the mitochondria are sitting, and the same happens with nucleus and the tubulin. When I apply the TauSense technology in Stellaris, what I get is out of 2 detectors, 4 lifetime-based channels. So now I can distinguish and I can identify the mitochondria and the actin filaments, and I can also see where my microtubules are and my nuclear stain. So this is enabled by a TauSense tool that is called TauSeparation. And I want to go into details of how it's implemented. So again, we are working with our sample, and we will get fluorescence intensity, and we can also get the photon arrival time. So again, working with this information at the FPGA, what I will have is a gate-based multi-component fit, and from this multi-component fit that works in the background and on the slide, the system will generate a diagram of the lifetime components. And that, added to a gating approach, will produce as an outcome lifetime-based separated channels. So there will be intensity channels in which I will be able to distribute the photons according to the species from where they come from. And these species differ in their fluorescence lifetime, so I can identify and I can separate them. In the example I showed you, I will have for the first detector then a lifetime-based channel 1 and a lifetime-based channel 2, and then I have the same for the second detector, and then I can distinguish the 4 different structures that I was labeling with different fluorescence lifetime molecules. So TauSeparation allows me to identify species with every image, even when their spectra are fully overlapping. So with this, I would like to close the potential of Stellaris chapter and the fluorescence lifetime-based aspects, and I will hand over again to Petra that will talk about the productivity of Stellaris.
So thank you, Julia. So now let's wrap that up by talking about the last pillar of Stellaris, which is productivity, and it's the productivity to do more. So Stellaris allows setting up complex experiments in a few clicks instead of setting up individual imaging parameters. And this also then results in less user training to get really to excellent results. So at the heart of doing more, there's something that we call the ImageCompass. And let me quickly tell you a little bit about the guiding principles that we had in mind when this was developed. So we wanted really to achieve that the user sample is in the center of setting up an experiment. So it should be able to set up without a lot of technical a priori knowledge an image and to get a high quality multicolor image really fast. Also, the interface should provide the feeling of full control to the user. So giving the feedback required to know exactly what's going on all the time. And finally, we wanted to reduce the complexity as a prerequisite to add new functionality. Basically, the functionality you just heard about from Julia when she was mentioning the TauContrast and TauSeparation features. So now let's have a look at how that actually works. So basically, the only thing you need to know is how the specimen under the microscope has been labeled. And based on this information, you pick a fluorophore from the available list of dyes and drag it into a setting. And the result is that each time a triad of excitation laser, spectral detection window, and respective emission spectrum is activated by the system. And also, what you can see is that an addition of a new dye always triggers an ad hoc optimization based on the new fluorophore scenario that you create by doing so. Stellaris also allows you to be fast across scales in time and space. So having the resonance scanner available by a single button click in the software gives you immediate access to high-speed imaging. Here on the left, you see a lifestyle imaging with an endosomal marker at 50 frames per second. If now we activate rolling average, image quality is improved big time while preserving the temporal resolution that is giving credit to the dynamic of your sample. So at the same time, raw data is always stored, so it's possible to revisit that to adjust it to the dynamic of your sample that you might observe only after the acquisition. So how does the rolling average work? Basically, once it's activated, it's applied also in live mode. And the more frames have been acquired, the better the image quality gets, basically up to the number of frames that you have defined. And from here on, the actual average starts to roll. During acquisition, the scheme is slightly different. Here as well, images get better all the way. Here in this example, up all the way to the 4th frame. And from there on, the actual image quality and display in the viewer gets slightly desynchronized. And in this example, again, when, for example, the 5th frame is displayed, the acquisition is actually already busy with the 9th frame and so on. So on top of that, to better adapt to the dynamics of the sample, as a user, you can control how much the remote frames are actually taken into consideration for the calculation of the central frame. So the different acquisition schemes that I have just introduced here are basically making sure that when you set up your experiment in live mode, you get already an estimate of how the final result will look like. Now, imagine that you can add the power of lightning to rolling average data set. In this example, you can see what you can achieve. So on the left side, you see raw data acquired with a resonance scanner, multicolor. And on the right side, this is a combination of rolling average. In this case, actually, it's rolling accumulation because the images have been acquired in counting mode, plus lightning processing. So there is a fully integrated workflow of rolling average with lightning and the resonance scanner. Last but not least, I want to mention our fantastic navigator tool, which will give you the ability to freely navigate through your sample and explore relevant details instantly in full quality. And by the way, this sequence shows an additional level of details made possible by TauSets. So the structural details that you can see here, the colors that arise from the different at the different structures are basically lifetime-based information which is revealed by TauContrast that was previously introduced by Julia. So Stellaris really gives you the productivity to do more. So Stellaris is offered in different configurations, including a broad range of modalities. such as, for example, STED, Falcon, DIVE, and DLS. And on this overview, you can see the differences between Stellaris 5 and Stellaris 8 regarding, for example, the availability of the power HIDE and also the modalities that I have just mentioned. Tausen, ImageCompass, Lightning, Rolling Average, and the Navigator are at the core of both microscopes. With this, we have already come to an end. I and Julia, we would like to thank you for staying all the way to the end. And now we would like to open up for live questions. Thank you. Thanks.
All right. Wow. Thank you so much. Those images were amazing. That was great. We did have a lot of questions come in, so we will get into those right away. If you still have a question you'd like to ask, please do so now. Just click on that Ask a Question box that is located on the far left of your screen. Okay, I'm going to go ahead and get to some of the questions that came in. The first one, for the NIR, is there a reduction of QE at the lower end of frequencies for detection, for instance, 488?
Okay, thank you very much for that question. It is, if you look at our power HIDE detector family, we have different types of detectors, as I had described. The HIDE R, so which is the dedicated detector for the NIR area, actually has a lower QE in this range of 488. So there you would not see a lot. On the other hand, if you look at the HIDE S detector, this is really a all-rounder, and you would see that it performs really, really well across the whole visible spectrum. So you would see a good signal from blue all the way into the near infrared.
Okay, excellent. Next question, let me just look at my list. Is this, let's see. It says, maybe you'll get to this, but I would love to know if, given you had a very bright tandem fret pair of CFP, YFP, how quickly can you capture a fret signal? My users work with cardiomyocytes, and we would like to use a tension sensor at high speed.
Thanks for the question. Actually, so it's a really great question, very interesting. So, of course, we can measure changes so based on the thousands tools very fast. And the main thing that one needs to consider here is how many photons you can collect. So the photon statistics, the photon budget. If this is a very bright tandem, then the next thing I would look into is the change that you would observe in your fret sensor, so when it's in one form and in a different form. So if there's enough difference, then yeah, of course you would be able to see it, and you would be able to capture it very fast, given that you have enough photons.
Okay, excellent. Let's see. Another one came in. Does the rolling average apply for highly dynamic objects that move faster than 10 to 20 micrometers per second?
I mean, yes, of course. I mean, if you work with the resonance scanner and you set that resonance scanner to an image format that allows you to have a high frame rate. So, for example, you can go up to something like 290 frames per second. And like this, you could, of course, also resolve these kinds of movements. And having this high frame rate also allows you to use rolling average on those samples to increase image quality while at the same time keep the temporal resolution.
Okay, excellent. Another one. How many photons do you need for TauSeparation to work?
Thank you. So we give an estimate of how many counts you need to find in each pixel. And as a rule of thumb, we would say you can have 50, so if you have 50 to 100 photons, that's a good starting point. So that would be the answer. So
okay,
at least 50 photons. Mm-hmm.
Okay. Next question: how fast can you acquire TauContrast images?
So as fast as your confocal imaging speed goes. So we can keep up the speed that you would do for your intensity-based imaging and get the TauContrast image. Of course, again, you need to consider here your photon budget. So.
Okay. Next question. Can STELLARIS microscopes be used for multi-photon CARS and SRS imaging modalities?
So yes, as you could see on this slide here, the STELLARIS comes in a STELLARIS 5 variant and also as a STELLARIS 8. And the STELLARIS 8 is really the platform that allows you to add all different modalities like multiphoton and CARS that have been asked for here. So yes.
Okay, great. Next, can you mix and match high D R and high D X to cover the full range of wavelengths without reduction in the QE?
I mean, yes and no. You, if you have a system where you can, where you have these different detectors available, then having the high DS and the high DR and the high DX is actually really good to get the best out at each individual location of the spectral range. But it's not that you have the same QE all over the full range. So you have a curve which is dropping towards the near infrared, but you still get very good signal out there.
Okay, great. Is the difference in photon counting leading to a large count rate?
The difference in, so that the fact that we have now the power counting enables us actually, yes, to collect more photons reliable. So we can go from really small count rates and have excellent signal there, but we can also go with samples that provide, that give more photons and be in a very good linear regime there as well, yes.
Okay. Next, do you need special probes to see at more red-shifted wavelengths?
Yes, so basically there are quite a large number, a nice number of commercially available fluorophores that now with this near infrared range, expanded range, we can very nicely implement in the labeling and then having the rest of the spectrum available. So yeah, so it's they are not exotic options. So those are things that we showed that you can purchase in different forms and apply it to your staining.
Okay. Okay. Next, how does sample depth and structural complexity affect the lifetime imaging?
Can you repeat the question?
Sure, yeah. How does sample depth and structural complexity affect the lifetime imaging? I apologize. If there's a typo in there, I'm not sure how to correct it.
Yes, so I am not so sure. So in principle, so so we can collect the arrival times of the photons. So even if the sample, I would say, even if the sample is very complex, we can, and if you image it properly and you sample it correctly, then you can get to those structural details and you can very nicely characterize it with the arrival time information of the photons. I'm not so sure if that is where this question is coming from.
Okay. Well, maybe that's when you can follow up with on email just to make sure that we've gotten it clearly. Yes. Sure. So this one's a little more straightforward. Can you use Alexa dyes or only proprietary fluorescent dyes? And what about photo bleaching with the Stellaris device?
So, of course, you can use any dye, so all dyes are welcome. So it's, so our confocal is suited for any commercially available or developed in your lab fluorophores. And what was the second part of the question? Oh, so yeah, I mean,
photo bleaching. Of course,
yeah, so of course, if you choose a good fluorophore, then you would get excellent results. And so in the way that we have implemented the white light laser, the detectors, and the optics, we made sure that you can image in fixed or in live specimens with very low laser conditions, and then you get excellent quality.
Okay. Next, does sorry, just lost my question there. Does TauSeparation get determined by the machine automatically?
Yes, so there's an integrated workflow and an algorithm that works at the background, and then you get these results automated. So you still can use this find function to determine the distributions where you want to set them or manually, but it's done automatically by the system.
Okay. Next, what if you stain for a protein in different cell types and the environment in those distinct cells change the way the actin shows different lifetimes? Wouldn't you get different colors for the same epitope?
Okay, so that's a long question. And so if you would have, I would say if you have a probe that when you use it in different cell types, then it reacts to the microenvironment differently, then you would get for the set spectral window, if you keep the spectral window constant, you would get your intensity-based measurement, and your color, let's say, would be the same, the corresponding to that spectral window. And what you will see is a change in the TauContrast based on the different response to the microenvironment.
Okay. This next one has two parts. Can Stellaris 8 have both WLL and 405 nanometer laser? And can it have 5 power S detectors inside and 4 more on the X1 port?
Oh,
if you want, I can ask them separately.
The Stellaris 8 can have, and as well actually the Stellaris 5, you can combine additional lasers to the white light laser, and one of these additional lasers can be also the 405 laser. What was the second part?
Can it have 5 power S detectors inside and 4 more on the X1 port?
So it can have 5 internal detectors. Yes, that's right. And for the X1 port, we currently offer 2 detectors, and those are AZTs.
Okay. And we've got time for one more. So last question, is there a light sheet option available with this technology?
Actually, yes. So we will have also on the Stellaris platform the possibility to offer our DLS, so the digital light sheet module, and this is available both on the Stellaris 5 as well as on the Stellaris 8, so on both machines.
Okay, well, thank you. Before we go, I'd like to thank you both one last time, and of course, thank the audience for joining us today. We also want to thank Labroots and our sponsor, Leica Microsystems, for underwriting today's educational webcast. All the questions that we didn't have time for today and those that will be submitted during the on-demand period will be addressed by the speakers using the contact information you provided at the time of registration. Julia or Petra, do you have any final comments you'd like to make?
Actually, yes. So I would like to say, and also in the name of Julia, thank you very much for taking part in this webinar. We hope actually that you enjoyed or that you had a good time. And if you have any additional questions that you did not answer now in this Q&A, just feel free also to contact your local Leica rep or contact us. So.
Okay, great, thanks. And for the
Thank you.
All right, and for the audience, this webcast can be viewed on demand. Labroots will alert you by email when it's available for replay. So we encourage you to share that email with any colleagues who might have missed today's event. And with that, I'll thank you one last time for joining and hope that you have a great day.